तो अ, फिर से आप सभी को रीड टू लीड चैनल में स्वागत है। आज हम कोशिश करेंगे कि 10 मिनट या फिर 20 मिनट या फिर 30 मिनट डेली एक बुक रीडिंग सेशन रखा जाए, क्योंकि बुक रीडिंग का आदत हमेशा होना चाहिए। एंड यही आदत मैं आप लोगों से बिल्कुल मैं चाहता हूँ कि आप में भी ये आदत मतलब बन जाए। और ये आदत तभी बनेगा जब सामने वाला आदमी भी करता हो। ठीक है। तो वाणी का प्रभाव कर्म से होता है, वाणी से नहीं होता है। वाणी का प्रभाव वाणी से नहीं आता है, वाणी का प्रभाव कर्म से आता है। ठीक है। तो हमने 29 1 26 में यहाँ तक मतलब कंप्लीट कर गए थे। ठीक है। एंड आज हम पढ़ेंगे टारगेट ऑफ ड्रग एक्शन। तो काफ़ी दिन मैं देख रहा हूँ हो गया है, इसीलिए मैं ये डेट सब मेंशन करता हूँ। ठीक है। मतलब कब पढ़ा था। ठीक है। तो यहाँ तक हमारा हो गया था। टारगेट ऑफ ड्रग एक्शन देखेंगे। टारगेट ऑफ ड्रग एक्शन नाउ, ये जो चैप्टर चल रहा है ये गुडमैन एंड गिलमैन का फार्माकोलॉजिकल बेसिस ऑफ थेराप्यूटिक्स। ठीक है। एंड चैप्टर 1 चल रहा है। ठीक है, आप देख सकते हो चैप्टर 1 जो कि ड्रग इन्वेंशन एंड फार्मास्यूटिकल इंडस्ट्री। ठीक है। चैप्टर 1 ड्रग इन्वेंशन एंड फार्मास्यूटिकल इंडस्ट्री। सो सीधा हम चलते हैं, यहाँ से स्टार्ट करेंगे। टारगेट ऑफ ड्रग एक्शन। Early drugs came from observation of the effect of a plant after their ingestion by animal, with no knowledge of the drug's mechanism or site of action. Although this approach is still useful example in screening for the capacity of a natural product to kill microorganism or malignant cell। ठीक है, malignant cell। थोड़ा स्टॉप वॉच को भी ऑन कर लेते हैं। malignant cell। Modern drug invention usually takes the opposite approach, starting with a statement or hypothesis that certain protein or pathways plays a critical role in the pathogenesis of certain disease, and that altering the protein's activity would be effective against that disease. Crucial questions arises. Can one find a drug that will have the desired effect against its target? Does, does modulation of the target protein affect the course of a disease? Does this project makes sense economically? The effort expended to find the desired drug will be determined by the degree of confidence in the answer to the last two questions. Is the target durable? The durability of a target with a low molecular weight organic molecule relies on the presence of a binding site for the drug that exhibit considerable affinity and selectivity. If the target is an enzyme or a receptor for a small ligand, one is encouraged. If the target is related to another protein that is known to have, for example, a binding site for a regulatory ligand, one is hopeful. However, if the known ligand are large peptide or protein with an extensive set of contact with their receptor, the challenge is much greater. If the goal is to disrupt interaction between two proteins, it may be necessary to find a hotspot that is crucial for the protein-protein interaction, and such a region may not be detected. Accessibility of the drug to its target also is critical. Extracellular targets are intrinsically easier to approach, and in general, only extracellular targets are accessible to macromolecular drugs. Has the target been validated? क्या ये टारगेट validated है? The question of whether the target has been validated is obviously a critical one. A negative answer, frequently obtained only retrospectively, is a common cause of failure in drug invention. Modern techniques of molecular biology offers powerful tool for validation of a potential drug target to the extent that the biology of the model system resemble human biology. Genes can be inserted, disrupted, and altered in mice. One can thereby create models of disease in animal or mimic the effect of a long-term disruption or activation of a given biological process. If, for example, disruption of the gene encoding a specific enzyme or a receptor has beneficial effect in a valid murine model of a human disease, one may believe that the potential drug target has been validated. Mutation in human also can provide extraordinarily valuable information. For example, loss of function mutation in the PCSK9 gene encoding proportion convertage subtilism obliquation type 9 greatly lower concentration of LDL cholesterol in blood and reduce the risk of myocardial infarction. Okay, based on these findings, two companies now market antibodies that inhibit the action of PCSK9. These antibodies lower the concentration of LDL cholesterol in blood substantially and are essentially additive to the effect of statin. Long-term outcome studies are in progress to determine whether the risk of a significant cardiovascular event also is reduced. Additional molecules are in the queue. Box 1-1 Target validation, the lesson of leptin. Biological systems frequently contain redundant element or can alter expression of a drug regulated element to compensate for the effect of a drug. In general, more important the function, the greater the complexity of the system. For example, many mechanisms control feeding and appetite, and a drug to control obesity has been notoriously difficult to find. The discovery of the hormone leptin, which suppresses appetite, was based on mutation in mice that cause loss of either leptin or its receptor. Either kind of a mutation result in enormous obesity in both mice and people. Leptin thus appeared to be a marvelous opportunity to treat obesity. However, on investigation, it was discovered that obese individuals have high circulating concentration of leptin and appear insensitive to its action. Is this drug invention effort economical, economically viable? Drug invention and development is expensive. See table 1.1. And economic realities influence the direction of a pharmaceutical research. For example, investor-owned companies generally cannot afford to develop product for rare disease or for disease that are common only in economically underdeveloped part of the world. Funds to invent drug targeting rare disease or diseases primarily affecting developing countries, developing countries, especially parasitic disease, often come from taxpayer or wealthy philanthropist. ठीक है. Additional preclinical research. Additional preclinical research. Following the path just described can yield a potential drug molecule that interact with a validated target and alter its function in the desired fashion. Now, one must consider all aspects of the molecule in question, its affinity and selectivity for interaction with the target, its pharmacokinetic properties, that is ADME, issue of its large scale synthesis or purification, its pharmaceutical properties, that is stability, solubility, questions of formulation, and its safety. One hopes to correct to the extent possible any obvious deficiencies by modification of the molecules itself or by changes in the way the molecule is presented for use. Before being administered to the people, potential drugs are tested for general toxicity by long-term monitoring of the activity of various systems in two species of animal. Generally, one rodent, usually the mouse, and one non-rodent, that is often the rabbit. Compound also are evaluated for carcinogenicity, genotoxicity, reproductive toxicity, see chapter number 4, in vitro and ex vivo assay are used when possible, both to spare animals and to minimize cost. If an unwanted effect is observed, an obvious question is whether it is mechanism based, that is caused by interaction of a drug with its intended target, or caused by an off target effect of the drug, which might be minimized by further optimization of the molecule. Before the drug candidate can be administered to the human subject in a clinical trial, the sponsor must file an IND application, a request to the US FDA, C clinical trial, for permission to use the drug for human research. The IND describes the rationale and preliminary evidence for efficacy in experimental system as well as pharmacology, toxicology, chemistry, manufacturing, and so forth. It also describes the plan मतलब protocols for investigating the drug in human subject. The FDA has 30 days to review the IND application by which time the agency may disapprove it, ask for more data, or allow initial clinical testing to proceed. Clinical trial. Role of the FDA. FDA is a federal regulatory agency within the US DHHS. It is responsible for protecting the public health by ensuring the safety, efficacy, and security of human and veterinary drugs, biological product, medical device, our nation's food supply, cosmetics, and products that emit radiation, sorry, Radi radiation. Okay, radiation FDA 2014. The FDA also is responsible for advancing public health by helping to speed innovation that makes medicine and foods more effective, safer, and more affordable, and by helping people obtain the accurate science based information they need to use medicine and foods to improve their health. New governmental regulation often result from tragedies. The first drug-related legislation in the US, the Federal Food and Drug Act of 1906, was concerned only with the interstate transport of adulterated or misbranded food and drugs. There were no obligation to establish drug efficacy or safety. This act was amended in 1938 after the death of over 100 children from elixir sulfanilamide, a solution of sulfanilamide in diethylene glycol, an excellent but highly toxic solvent and an ingredient in antifreeze. The enforcement of the Amendment Act was entrusted to the FDA, which began requiring toxicity studies as well as approval of NDA. See the conduct of clinical trials before a drug could be promoted and distributed. Although a new drug safety has had to be demonstrated, no proof of efficacy was required. ठीक है, थैंक्स फॉर वॉचिंग. 15 मिनट हो चुका है. आज का डेट कितना है? आज का डेट 15 फरवरी, 15 फरवरी 26. टाइम 3:29 PM. And thanks for watching.